Today we're discussing small peptide affinity tags such as his tags. These small peptide tags help us purify proteins using affinity purification. The affinity or attraction between the binding partner and your tag target protein helps draw that target protein out of solution away from other contaminating proteins. In order to purify the protein that we want using affinity purification, we need those small peptide tags. Let's imagine being at baggage claim at the airport for a moment, and maybe you're traveling with a sports team, so all the luggage coming around looks exactly the same. What's going to help you find your specific suitcase is the luggage tag, the one with your name on it. This is kind of how affinity purification works. In the lab, we'll attach a small peptide tag to a protein of interest, which is like putting the luggage tag on our suitcase. Then we'll run a sample through the purification system that recognizes and grabs only the tagged protein, and everything else will just wash out. But there are different types of small peptide tags with certain advantages. So we're looking at 3 of the common ones today. That's his tag, flag tag, and Strep-tags. The his tag is short for a histidine tag. This is a sequence of 6 to 10 histidine residues that bind strongly to nickel and other divalent metal ions. With a his tag, you can perform purification using the immobilized metal affinity chromatography method, or IMAC method. What happens is that when the his tag is passed through a column that contains nickel, the tag sticks to the nickel and everything else washes away. And because your protein of interest is attached to the his tag, you've also captured your protein of interest. Once the other unwanted proteins are washed out, you can elute your protein using a competitor like imidazole in this case. The his tag method is really fast, efficient, and very widely used in labs. The next tag on our list is the flag tag. Unlike his tags, which use metal interactions, the flag tag relies on antibodies that recognize the flag tag and therefore detect the tagged protein. For this reason, flag is a good alternative tag for proteins that are incompatible with his tags because they bind to metals or are destabilized by high concentrations of imidazole. When purifying your proteins using a flag tag, your flag tagged protein is loaded into a column with a specific flag recognizing antibody conjugated to the aggro speed. Usually that's going to be the M1 antibody, M2 antibody, or the M5 antibody. The antibody is going to recognize the flag tag and capture it, and therefore, again, capture your protein of interest. The column then gets washed, so your undesired proteins are washed out. Then you would elute your protein using more flag peptides to disrupt the interaction between the flag tagged protein and the antibody labeled beads. Our third and last small peptide tag is the strep tag. A strep tag is a short peptide sequence that binds to the bacterial protein Streptavidin with really high affinity. The strep tag is a really good choice when only one purification step is going to be used. Because of the high affinity between the tag and Streptavidin, eluted proteins are generally really pure compared to other tags like his tags. When you're working with strep tags, you would have your strep tagged proteins loaded onto the column with your Streptavidin conjugated aggro beads. These Streptavidin beads are going to capture the strep tag and, of course, therefore your protein of interest. So just like the other tags, your unwanted proteins can wash out. And then after that, you would just elute your protein of interest. So if you just want to do one purification step and be done, using strep tag is a good choice. Finally, I just want to go over some important tagging considerations. Affinity tags are often used in protein purification, and they're really user-friendly. But there are 2 really important things to think about to make sure that you have more success. First, you want to ensure that the affinity tag you're using doesn't interfere with the protein's function. And second, you'll want to decide whether to keep the affinity tag or get rid of it for downstream applications. To elaborate on these points just a little more, let's talk about the affinity tag and a protein's function. Usually these tags really aren't going to hurt your protein when they're added. They also don't usually interfere interfere with a protein's function. But you'll want to verify that. And if the tag does interfere with your protein's function, that might mean that the tag is interfering with a protein's fold or that it binds to an important interface on the protein's surface. When the small peptide tags are added, they're added to a protein's amino or carboxy terminus, or more rarely, within a protein's flexible loops. If you discover that the tag is interfering with a structure or function, you can try moving it to another location and then retest. Alternatively, if you have one affinity tag that is interfering with your protein's function, you can try swapping it out for another one. Different tags have different properties. Flag tags are positively charged, strep tags are relatively hydrophobic, and his tags are just histidine. So just because one tag interferes with your protein's function doesn't mean that another one will. For the downstream considerations, when you're designing an expression construct for fusion proteins, you want to think about whether the tag is tolerable for your downstream uses. Certain assays do require tagged proteins, especially ones that measure molecular interactions. So in these cases, when you're purifying your proteins, you would want to keep your affinity tag on your protein of interest. But if you don't need to keep it for your downstream processes, you could add a protease cleavage site between your affinity tag and your protein of interest. The cool thing is that with a flag tag, that sequence already contains a protease cleavage site within the affinity tag. But a lot of the other tags, including your his tag and strep tags, would need that cleavage site added. So that's the breakdown of some of the common small peptide tags out there. If you're doing protein purification, GoldBio has a lot of other resources and articles to help you. And not just resources, GoldBio's website has a lot of reagents that are quality tested and really affordable for protein purification. Hey everyone, just really quick, if you're interested in other resources to help you along, you might want to check out GoldBio's handbooks. We have all sorts of different handbooks addressing topics like lab management, science writing, and we also have more specific books for types of products and methods like PCR, growth factors, luciferin, and biological buffers. Check out the link in the description to browse and download.